In 2021, a geneticist wrote 3 words in his lab notebook: "Check the numbers again." Not because the equipment had failed, not because the software had crashed, but because what he was looking at did not fit any model his team had built going into the study. He was staring at a comparison that had never been done at this scale before. Black American DNA versus Chinese American DNA. Side by side, millions of genetic markers, 50,000 participants, months of processing time on some of the most powerful sequencing technology on the planet. And the results that came back challenged something that American medicine had been doing for decades. Not just how we think about race, how we treat patients, how we prescribe medications, how we calculate disease risk, how we make life and death clinical decisions based on a category that the DNA data kept refusing to validate. When these findings were published, a researcher at Johns Hopkins said something that got quoted in 3 separate medical journals. "We have been measuring the wrong thing and calling it biology. This is what the data actually showed." If this is the kind of content you want every week, subscribe right now. I cover genetic discoveries that are still years away from making it into textbooks. Now let us get into what the science revealed. To understand why this comparison was so significant, you need to know what motivated it in the first place. For decades, American medicine used race as a clinical variable, not as a social descriptor, as a biological input. When doctors calculated kidney function using the EGFR equation, race was a factor in the formula. Black patients received a different calculation than non-black patients, which often resulted in delayed referrals to specialists and worse outcomes. When cardiologists assessed heart disease risk, race-based adjustments were built into the risk calculators. When pharmacists considered drug dosing, population-level assumptions about metabolism were often tied to racial categories. All of this rested on one foundational assumption: that racial groups are biologically distinct in ways that are medically relevant, that black patients share enough genetic similarity with each other and enough genetic difference from other groups that race functions as a useful proxy for genetic information. Nobody had tested this assumption directly against actual DNA data at scale. That is what the 2021 study set out to do. Researchers at Johns Hopkins and the University of California, San Francisco recruited over 50,000 participants. Black Americans from across the country, Chinese Americans from major metropolitan areas on both coasts and in the Midwest. They sequenced millions of single nucleotide polymorphisms, the specific positions in the genome where individuals differ from each other. They mapped haplogroups, the deep ancestral lineage markers passed down through maternal and paternal lines. They identified disease-associated variants. They analyzed drug metabolism genes. Then they ran the comparison. And the first result came back before the main analysis even finished. When researchers analyzed the genetic diversity within the Black American sample alone, the numbers stopped them cold. This population was genetically diverse to a degree that immediately complicated the study design. The internal genetic variation within Black Americans was so wide that the standard statistical models researchers planned to use had to be rebuilt before meaningful comparisons could run. This was not entirely surprising to population geneticists. It was, however, far more extreme than clinicians who designed race-based medical protocols had ever accounted for. Here is why it happened. Black Americans are not the descendants of a single African population. The transatlantic slave trade forcibly transported people from dozens of genetically distinct West and Central African ethnic groups to the Americas. Yoruba from what is now Nigeria, Akan from Ghana, Igbo from southeastern Nigeria, Wolof from Senegal, Bakongo from Central Africa, Fawn from Benin. These groups had been genetically diverging from each other for thousands of years before the slave trade began. They had different haplogroup distributions, different disease-associated variant frequencies, different metabolic profiles. Slavery compressed them into a single legal and social category. Their DNA did not compress. Then there was the genetic mixing imposed by slavery itself. Decades of genetic research consistently show that Black Americans carry on average between 15 and 25% European ancestry. Some individuals carry more, some carry less, but the result of centuries of forced reproduction during enslavement added yet another layer of genetic complexity on top of the already diverse African foundation. What this created was a population whose genetic spread was extraordinary. The Chinese American sample showed the opposite pattern: relatively low internal diversity, tightly clustered genetic profiles, clear Han Chinese ancestry with minimal admixture from other populations. The historical reason was straightforward. Most Chinese American families traced their immigration to Guangdong and Fujian provinces, because those coastal regions were the primary sources of 19th and early 20th century emigration to the United States. Han Chinese ancestry itself reflects thousands of years of relative genetic stability within China, which population genetics research has consistently documented. So researchers were now looking at two populations with fundamentally different internal genetic structures. One with enormous diversity built from forced migration and the merging of dozens of African ethnic lineages. One with much lower diversity built from voluntary migration from a geographically concentrated source population. Now here is the finding that created silence in the lab. When the cross-population comparison ran, one result came back that the team had not predicted. The genetic distance between Black Americans and Chinese Americans was smaller than the genetic distance within the Black American population itself. Read that again slowly. Two Black Americans from different parts of the African diaspora could be more genetically distant from each other than either of them was from a Chinese American individual. This was not a statistical edge case. It reproduced consistently across thousands of sample pairs. What this meant was direct and uncomfortable. Black Americans as a group do not form a genetically coherent category in the way that medical protocols had assumed. The label Black American describes a social reality, a historical reality, a lived experience shaped by centuries of racism, discrimination, and shared cultural identity. But as a genetic category, it encompasses so much internal diversity that treating it as a biologically uniform group produces medically inaccurate results. And this is where the study moved from being scientifically interesting to being clinically urgent. Because if the genetic variation within Black Americans exceeds the average genetic distance between Black Americans and Chinese Americans, then a doctor who assumes all Black patients share the same genetic disease risk profile is not just making a rough approximation. They are working with a model that the data does not support. The implications for how medicine is practiced in the United States were immediate and unavoidable. The research team moved through specific disease categories and the findings accumulated. Type 2 diabetes first. The genetic variants most strongly associated with diabetes risk in European-derived populations showed up at completely different frequencies in both Black Americans and Chinese Americans. That much was expected. Populations with different ancestries carried different variant distributions. What was not expected was the degree of variation within the Black American sample itself. A Black American patient, whose ancestry traced predominantly to Yoruba lineages from southwestern Nigeria, carried a different constellation of diabetes risk variants than a Black American patient, whose ancestry traced predominantly to Akan lineages from Ghana. Both patients would receive identical race-based risk assessments under current clinical protocols. The DINA said those assessments were not equivalent. Cardiovascular disease showed the same pattern. Specific variants linked to heart disease risk, variants in genes affecting cholesterol metabolism and arterial inflammation, were distributed so differently within the Black American population that group-level risk estimates had poor predictive accuracy at the individual level. Drug metabolism was the most operationally direct finding. The genes that control how quickly the human body breaks down common medications, CYP2D6, CYP2C19, CYP3A4, these showed significant variation both between the two populations and within the Black American population specifically. These are not abstract genetic curiosities. These genes determine whether a standard dose of a medication produces the intended effect, causes toxicity, or does nothing at all. A patient who metabolizes a drug slowly needs a lower dose. A patient who metabolizes it quickly may need a higher dose or a different medication entirely. Race-based dosing assumptions collapsed when tested against actual variant data. The researchers calculated that individual genetic testing outperformed race-based clinical assumptions in predicting drug response by a significant margin in both populations, but especially in the Black American sample where internal diversity made group averages least reliable. The conclusion the team published was direct. Race-based clinical medicine is not just scientifically imprecise. In specific contexts, it produces measurably worse patient outcomes. And the population most harmed by this imprecision is the population whose internal genetic diversity makes group averages least applicable to individuals. One more finding emerged from the deep ancestry analysis that the team had not set out to find. When researchers traced the deepest genetic layers in the Chinese-American sample, they found markers that followed far enough back through human evolutionary history connected to African populations, not through any recent contact or admixture, through shared ancient origin. The haplogroups that dominate East Asian populations today, mitochondrial haplogroup M and Y chromosome haplogroup O among others, are themselves descended from African ancestral haplogroups. When small founding populations left Africa between 60,000 and 70,000 years ago, and moved eastward across the Asian continent, they carried those haplogroups with them. Over tens of thousands of years, those populations differentiated. They developed distinct physical features. Their genomes accumulated different mutations. Their disease profiles diverged. They became, in every practical sense, genetically distinct from the African populations they had descended from. But the shared origin is encoded in every genome. Population geneticists have calculated that approximately 7,000 years ago, every person alive today with living descendants is an ancestor of every person currently alive on Earth. The mathematics of genealogy guarantees it. Family trees expand exponentially going backward, and the total number of possible ancestors quickly exceeds the actual population of humans who existed at any given time in the past, which means everyone's ancestry overlaps. The Black American family tree and the Chinese American family tree converge at a point in the relatively recent past. The researchers were careful about what this meant and what it did not mean. It did not mean the populations were genetically the same. The differences in disease risk, ancestral haplogroup distribution, and drug metabolism were real, and the study had documented them extensively. What it meant was that the deepest structure of human genetics does not recognize the racial categories we imposed on top of it. Those categories have enormous social meaning. They have shaped access to resources, opportunity, safety, and power in America for centuries. That social reality is not diminished by the genetic findings. But as a biological classification system, race was always doing something different from what the people who built medical protocols around it thought it was doing. When the study was published, two responses came back from the medical community. The first was relief from researchers who had been arguing against race-based medicine for years. The data at this scale, with this sample size, using this methodology, gave them the clearest evidence yet that the foundational assumption underlying race-based clinical protocols did not hold up. The second response was more uncomfortable. If we know this, why is race still embedded in clinical calculators that are used in hospitals every day? Why was the kidney function equation that disadvantaged Black patients only revised in 2021, after years of advocacy from nephrologists and patient groups? Why are drug trials still stratified by race in ways that assume genetic uniformity within racial groups? The researchers did not argue that the solution was to ignore population-level genetic patterns entirely. Those patterns are real, and they have medical relevance. The argument was that race is too crude an instrument to capture those patterns accurately. Individual genetic testing does what race-based medicine tried to do but could never actually accomplish. It tells you specifically which variants a specific patient carries. It tells you exactly how that patient metabolizes a specific drug. It tells you which disease risks are genuinely elevated for that individual, based on their actual genetic architecture, rather than based on how they look or how they identify racially. That future is already arriving. Pharmacogenomic testing is increasingly available. Ancestry-specific risk modeling is being built into clinical tools. The gap between what genetics can tell us and what race-based medicine was approximating is closing. But getting there requires first accepting what the data showed. When scientists compared Black American to Chinese American at scale, the results shocked everyone, not because they revealed that the two populations were the same, but because they revealed that the category used to call them different was never measuring what medicine assumed it was measuring. The DNA was always more complicated than the label. It always will be.